Hello everyone, this is Dhanashri from the Dr. Omics lab. In this topic, we are going to start the CRISPR data analysis. Basically, it's a CRISPR-Cas9 system, which stands for clustered, regularly interspaced, short palindromic repeats, and the CRISPR-associated protein 9, which is a Cas9. Basically, this is a genome editing technology that has revolutionized molecular biology due to its precise and a site-specific gene editing capabilities. It is widely used in a genetic research, biotechnology, and the medicines, and that's why we are going to discuss over here that how this CRISPR will work, how it will work mechanically, what are the different applications, then how we are going to perform the data analysis. Everything we are going to discuss very detailedly in the upcoming part. But before that, let's understand some history of the CRISPR. In 1987, Atsuko Nakata and its team of researchers from Osaka University in Japan first reported the presence of clustered, regularly interspaced short palindromic repeats, which is abbreviated as a CRISPR, and it is discovered in a Escherichia coli or E. coli genome. This refers to short, repeated sequence of DNA nucleotide which is found within the genome of prokaryotes. Now, what is a palindromic? Palindromic sequences means what? Like these sequences are same when read from 5 prime to 3 prime on one strand of a DNA and 5 prime to 3 prime on a complementary strand. Means like whenever you are seeing from a both side, it is looking same only. That's what we call it as a palindromic sequence. Then later on in mid-2000, the functionality and the importance of CRISPR was first realized in prokaryotes. As it turns out, the CRISPR system is a key component of their adaptive immunity, which protects these prokaryotes from attack by viral DNA or a bacteriophage and a plasmids. Vaccination is one of the best example of adaptive immunity, you can say, which is results in the form of adaptive immunity in humans, since the body is exposed to the antigens and forms antibodies in response, which contribute to the development of the immunity. So the unique sequence that are nestled in between the palindromic repeats, that we call it as a spacers. Means in these organisms, this CRISPR is used to define themselves against the virus, or you can say the bacteriophage. The CRISPR system works by remembering the viral DNA sequence from a previous infections, which is allowing a bacteria to recognize and cut the DNA of invading virus in a future encounters. So that's why whenever we are talking about the CRISPR, there are two main key components. One is the repeats, another is a spacer. So repeats are nothing but what? It's a short palindromic DNA sequence that are identical and separated by the spacer DNA. And spacers are nothing but what? These are the DNA fragment from a virus the bacteria have encountered, which is act as a memory for that viral infection. So this is a memory sequence, you can say. But actually, there are another two main other components we need to be understand to learn the CRISPR, which is the Cas9 and the guide RNA. Cas9, also called it as a molecular Caesar, which is a CRISPR associated protein 9. That's a Cas9. It is an enzyme that functions as a pair of molecular Caesars. It can double-stranded the DNA at a specific location, which is determined by the guide RNA, which is gRNA, and the cutting mechanism is the basis of gene editing. And the next is what? It's a guide RNA. So basically, guide RNA is a synthetic RNA molecule that is designed to match the target DNA, and it directs the Cas9 protein to specify like for the specific location in the genome that needs to be edited. How this CRISPR will work? Actually, it's a simply two-component system that allows researcher to precisely edit any sequence in the genome of organism. This is achieved by the guide RNA, which is recognized in the target sequence, and the CRISPR-associated endonuclease, that is Cas, that cuts the target sequence, and the activation of Cas9 protein by the guide RNA binding. So this binding of guide RNA induces the conformational change in the Cas9 protein, and the conformational change causes the activation of Cas9 endonuclease. If I will just explain this diagram, which is given over here, so to understand it better, I will just paste it here in the paint. Yeah. So here in this diagram, if you will see that this is like it is showing at its core the CRISPR involves the guide RNA, gRNA. You can see here. This is what a guide RNA here it is showing, which is showing in the green color. And that leads a Cas9. Here, this guide RNA, you can see that it is attached with the Cas9 enzyme. It is binding with the Cas9. So it leads the Cas9, which is a cutting enzyme, to the specific part of the DNA. The guide RNA matches a target DNA sequence and binds to it. So the guide RNA ensures that Cas9 is brought exactly where the cut needs to happen. Just next to the target DNA sequence, there is a one small motif we are having, which is a PAM sequence. So this is here, which is given is nothing but the PAM sequence given in a red in color, which is necessary for Cas9 to work. Like this PAM sequence is one of the known sequence where your Cas9 is going to bind, and along with the PAM sequence only you are having your target DNA. So once the guide RNA finds the correct DNA, Cas9 acting like a molecular Caesar, and it is going to cut both the strands of the DNA at a catalytic center, which is marked as a bled over here. You can see that. So this allows for modification to be made to the DNA either by disabling a gene or inserting a new genetic material. So what we are going to do after that, like once this is done, once the cutting is done, here you can see there are two mechanisms are working over here, which are like a repurposing the CRISPR-Cas9 system for genome editing, which is exploits the DNA repair mechanism of eukaryotic cells. And there are two repair mechanisms are there. One is a non-homologous end joining, which is NHEJ, and another is a homology-directed repair pathway. So this non-homologous end joining pathway, which is NHEJ, this process is error prone and often leads to point mutations, deletions, or cause like frame shift that alter the gene product and eventually abolishes its function, which is a favored for genetic knockouts. And HDR, this is a precise genome engineering, like it's relies on pathways. Here, a piece of DNA that shows a sequence homology to the target site and is used to repair the DNA via homologous recombination. This short DNA sequence can harbor any sort of insertion or alteration, so it's allowing the integration of any desirable DNA sequence at the target site. So once this DNA repair mechanism is done, after that, whatever the insertion, deletions, or a point mutations, or any kind of variation, if we want to find out in this DNA, so for that we are going to perform the CRISPR data analysis. So basically for that, we need to follow some steps, we need to follow some pipeline. Like the DNA, we need to perform the sequencing for that, and once the sequencing is done, we need to check the quality for it, like quality control we need to perform, and for that we are going to use the fast QC. Maybe if the data is not having a good quality, we are going to be processed for the trimming, and for the trimming we are using the FastP. So once the trimming is done and now you are sure that yes, your data is having a good quality, then you can directly perform the alignment, like aligning of reads to the reference genome. Because like if you will see that here, what is the main thing? Once you are having a raw data or once you will get the sequence of the DNA which is edited, so you need to, let's consider this is what a raw data which is having some reads inside it. So those reads you need to be matched against or you need to align against the reference genome of the organism. Let us consider this data is from the Homo sapien. So you just need to be align those reads against the Homo sapien reference genome. And we already know from the Homo sapien reference genome the bases are present. So let us consider like you are aligning the reads here, and if in the reference it is showing you A, and the reads which are aligned over here it is showing you some another base like G, or maybe it is showing some insertion over here or some deletion over here. So that thing we are going to detect as a mutation, and this is what we are going to perform in a CRISPR data analysis, and that's why we need to align the reads against the reference genome. So after the alignment, next we are going to perform the variation calling, and for that we are going to use the GATK. So by the variation calling, we will be able to understand at any specific position, is there any point mutation, is there any insertion, deletion, or any kind of variation is there or not. So this is how basically we are going to be processed for the CRISPR data analysis. Our aim over here is to find out the variations caused by the gene editing, and that's what we are going to process in upcoming part. So here if you have any question, please let us know. Till then, bye. Have a great day.